Okay.
Tenuous cephalus.
Awesome.
See, I knew it started with
Thank
T.
you. Yeah.
Oh.
So one of the questions coming in from the chat is, do we find trash on the sea floor? And the answer is yes. Actually, most dives we do.
Did they see trash on the last time?
We did not.
No.
So that was an exception.
Yeah.
And I don't think we
None
have on this one.
No, not yet.
Yeah.
But you see things like fish nets,
We're
sometimes
pretty
pop
remote
cans.
right here.
We are
Yeah.
pretty remote.
Wow.
A lot of plastic sometimes.
Yeah. I've seen pop cans from the 80s down in the Lao Basin.
Wow.
I've seen a can of Spam.
Wow.
Spam. Oh my god.
It was 25% less sodium.
Oh, the light spam.
Less sodium than pure salt.
Yeah, well, not anymore. It's
Less
100%
salty than
sodium
the
now.
sea. So yeah, here we're seeing lavas that look in place. They're pretty heavily encrusted, and it looks like this crust has been growing for quite some time.
And what evidence shows that it's like probably really stuck together? What are you seeing on there?
I'm seeing a lot of very similar crustal morphology on the rocks to each other, so they've kind of got that planar look to them with that edge running around somewhat horizontally.
Yeah.
They're all very similar in that aspect, and it's all oriented about the same way from across different parts of this this outcropping here. So that tells me that all that's been growing in place for probably tens of millions of years.
It can be very deceiving. It'll look like it's like just sitting there on the surface, and then you go to nudge it, and it's connected to the planet.
Yes, it sure is. And I believe that is exactly what we're looking at right here. So some of these pieces of rock sticking out of the sediment, they look like cobbles that are just sitting there. My guess is they are part of the seafloor now.
Yeah.
Wow.
Either in place or debris that fell so long ago that it's crusted over and glued back down. Yeah, so this isn't a good place to grab a rock. I'm more interested in getting into
There's a little
rock.
something on that rock.
Yeah, so we're starting to see some things looking a little spongy, maybe.
Video, can you zoom in?
Copy, zoom in.
I think it might be a scallop.
Oh.
I think you might be right.
Wow.
So Megan's doing a lot of our ID. She's looked at so much footage from so many of these dives that she's got really keen eyes for all of this. So
Yeah.
Megan, I'm relying on you.
Yeah, these are definitely scallops in the family Pectinidae.
Oh, wow.
So bivalves, perfect, very cool.
Is that a smaller one next to the
Yeah,
big
little one.
Yeah.
Oh, wow.
So it looks like they only have
This picture will make it into the guide.
So these don't look totally symmetrical. They've got
They're
a
not,
one side
yeah.
of the hinge is pointier than the other.
I've never
That
seen
happens
that.
a lot in a lot of scallops, where
Okay.
they're
Oh.
not perfectly symmetrical.
Okay.
Oh, wow.
All right, carrying on.
Carry on.
Carry on.
Coming wide.
Bonk. Oops.
So that's interesting. This is the second dive that we've had in this part of the world where we've seen just oodles of sediment.
Yeah. Does that tell like a geologic history there, or more like an ocean current kind of information, or both?
I think it's more of an ocean current story.
Yeah.
We'll see, you know, as we gather more data from more seamounts, that'll start to put together more and more of the picture.
Oh.
It's hard to say much right now, but it looks like, yeah, more sediment deposition in this area. Beyond that, don't know yet.
Yeah.
Annette?
Yes?
Yes. Pump 2, 81871. Pump 3, 79765. Thanks.
So here in the ferromanganese crust, you can see that some of the surface texture looks like it has more parallel lines to it, and that's sort of a long-term indicator of current directions.
Wow.
So the way the water's moving is having some influence on the surface morphology of the crusts.
Mm-hmm.
Over a long period of time, too, right?
Very long, yeah. Like millions of years. It's prevailing winds.
Wow. And what causes manganese crust to form over these rocks?
So it precipitates out of seawater.
Oh.
And there's not a whole lot of iron or manganese, the main constituents of ferromanganese crusts, or the other trace metals that accumulate in those. There's not a huge load of those dissolved in seawater, so it's a very, very slow
It's an
growth.
anemone.
Anemone.
I think somebody's getting hungry.
You zoom in?
Right, zooming.
Ooh. Oh, look at that.
And some of these anemones actually will root in sediment.
Looks like that one has, yeah.
Oh, wow. So what kind of organisms would this sea anemone feed on?
Usually small crustaceans, polychaetes, those types of things.
Oh. That's so cool.
Right, very nice.
There we go.
All right, so we're pretty much on top of waypoint 3 right now and making our way toward waypoint 4. And we have, the moment, 9 total waypoints in this dive plan, but once we get further up slope, we're hoping to see a higher population density of individuals and colonies around here. That's the hope anyway, so we might take more time further up slope between some of these waypoints if that's the case.
Yeah.
Yeah.
Oh, wow. That's a big one.
I like that rock.
It's so
That's a
strange.
nice rock.
It's huge.
It won't fit on the ROV though.
ROV will fit on it.
You could try.
Wowie.
Oh, wow. That thing is massive.
Mm-hmm. So if there are a little less sediment coating the surface of this would be one of those outcrops where if the current and the nutrient load in the water column was good enough, we would be seeing a ton of corals on. But this is kind of sparse, extremely
We're not
sparse.
to the top yet.
Yeah, yeah.
We've got a long way to go to get to the tippy top.
At the top of the rock. The local high.
Mm-hmm. Yeah, sometimes you see stuff hanging out on the sides though, but yeah, with the sedimentation around here, I'm not sure that that's helping the matter,
Yeah.
because that can kind of choke some of the filter feeders like the sponges. And it's a little harder to root yourself to the substrate with sediment on top of it. So most of this looks in place. All of it looks stuck. So Not looking for a rock in this area.
Ooh, we have a good question. Somebody asked Megan, what is your background and what you study?
So my background is in biology. I did my undergrad and graduate studies as a marine biologist. But as a biologist in the marine environment, we study a lot of everything. We have to understand the geology, the chemistry, and the physics of the ocean in order to really understand the biology that we're seeing and observing. My thesis research was on the development and community structure of deep sea corals off the Big Island of Hawaii. I used the ages of the lava flows to estimate the age of a coral community and try to piece together how those coral communities change over time, seeing as it's really hard to study these slow-growing animals over a human lifetime, so we needed to use something a little more geologic as a proxy for what we'd use on shallow water coral reefs called coral settlement plates, which we can put out and see how the corals will come and colonize a plate and piece together how the community succession works. And we use the lava flows for that. And it was really cool. Found that these communities are very, very old. It takes a very long time for them to develop, which we anticipated, seeing as we know a lot of these corals grow very, very slowly and live extremely long lives, some thousands of years.
Wow.
That's amazing.
Ooh, we are seeing some dead sponge stalks here.
Oh, is that
Perhaps a hint of what's to come. Maybe or maybe not.
the teaser.
We'll find out. Maybe. All right, so there is a question about where we are. So we're toward the center of the Marshall Islands Exclusive Economic Zone, or EEZ. We're over on the west of the two main subchains comprising the Marshall Islands, known as the Rollick chain. And we are slightly, we're like north-northwest of Kwajalein Atoll. And let's see, this is on a seamount called Luanjua. I think I have that pronounced, I think I'm pronouncing that almost correctly. It's a little hard with my accent. So yeah, we're sort of in the central Marshall Islands region right now.
Oh.
And if we, it also looks like, yeah, there might be a little update issue with the location on the website. So I'll pass that along to our data person. And we'll get that checked up on.
It's getting a little breezy over here.
Is it?
Is it?
Yeah.
What direction is
in my pilot seat.
Oh, not the current.
You're feeling it?
Okay.
Yeah.
We're taking control of it
Hour
this time,
3
not
where you're
her.
like, a little bit,
I'm not appropriately attired for...
Yeah, it does get chilly in here.
It does.
Our poor videographer sits right under the fans, so she's bundled up.
Yeah, and I also have a puffer jacket, a sweatshirt, and a blanket, and a heater warmer.
Yeah.
The blanket combo's where it's at.
I left your blanket over there. Oh, you got it. Okay, good.
Yeah.
I
Blanket.
definitely brought my blanket in here before.
I'm not wearing a blanket. Oh, you did? How nice. Thank you. Oh, wonderful. Now I'll be more comfortable.
We should
Thank you.
have some emergency blankets in here for...
Right?
I have an
Right.
extra puffy blanket in my stateroom just in case anybody needs it.
Ooh.
We should just have
just
them
a little
available in here, though.
I was saying we
might
should get
get
like
out of
team
control.
parkas, you know?
Ooh.
Or snuggies. That's
A
nice.
Nautilus
Ooh,
blanket
snuggies.
nest.
The
Yes.
best.
But the
I like
best
the
have
snuggie
arms.
idea.
Yeah.
My arms get cold.
Still, but it still makes a big difference having a vest on.
It does help. Oh no, my bottom gets a little chilly.
They did give us some sweaters once. They're kind of itchy though. I'm kind of particular about my sweater types. So.
Yeah.
Me too.
I think we need like Nautilus hoodies.
Yeah. We haven't done that before.
Hoodies would be so awesome.
There are Patagonia sweaters. They're just kind of, that's kind of scratchy kind of stuff.
No, it has to
Oh,
be
those
soft.
could be itchy sometimes.
This is
Critter.
a nice critter,
Ooh.
nice sea cucumber. This is
And
a
there's an anemone too.
Peniagony or an Amperima.
That's
We
a
did
large
that
one.
video?
Wait,
Sammy.
wow. Oh, look at that.
And it has
So
a
these
friend.
sea cucumbers can swim. They
Do
use
they
that.
discharge ballast when they...
They usually do discharge some ballast before they take off.
Got to maintain that buoyancy, right?
Yeah. And what is that? Like, is it a fin on the back of it?
Yeah,
Yeah.
so that's called their velum, and they can use it to push water and swim when they do their sea cucumber crunches.
Oh, how cute.
Yeah, they just do their crunches and they just swim through the water column. It's really fun.
Oh, to be a sea cucumber.
It would be an interesting life.
It would be.
Yeah, usually some of these will take off when we get close to them. They feel the vibrations from the ROV and they'll jump.
They're just like,
Oh.
yeah,
So this
no,
one's in the
we're not doing this.
thinking
Yeah.
about jumping.
Yeah.
Oh.
It's just going to nope out of there.
There it goes.
Oh, is it getting
No.
ready?
Oh, maybe not. Maybe
Getting
it
ready
changed
for
its mind.
takeoff?
It's very comfy there.
No, it's not going to swim for us.
Can't be
No.
bothered.
It's really happy where it is.
Yeah.
All right, carry it on.
Ooh, we had a question just come in from somebody. They're asking, what is the chance of spotting any squids at the depth we're in right now?
Squids?
Yeah.
Vampire squids, maybe, huh?
Oh,
Don't those show up on abyssal plains sometimes?
do they? I know we have the squids when Hercules is being pulled up or down, right?
Yeah.
With the
blue
lights.
water.
So what's the word on squid?
Well, what was the question?
Oh, someone was asking, what is the chance of spotting any squids here?
I mean, it's not impossible, but it's improbable.
Mm-hmm.
We might see a Dumbo octopus.
Yeah, we do
That'll
see
be
more
fun.
octopus at depth.
Yeah,
Oh.
but it's rarer to see the squids. They're very fast. They know we're here.
Yeah.
Oh.
They're
And
around, but they definitely avoid us.
we've been told to keep eyes open for Casper octopuses.
Oh.
Yeah, we aren't
Yeah.
deep enough that it's a possibility to see one of those, but those
Yeah.
are also very, very rare.
Wow.
Yeah, I think we saw one on 154, and that's the only time I've ever seen one.
Yeah,
but I haven't
that was like the
Really?
second time it had been seen.
Yeah.
Oh, wow. Oh my
No
gosh.
way.
But that was up in the monument, in the Papahanao-Mokuakea Marine National
Yes.
Monument.
We were on watch when we saw that, Val. It
We
was
were,
amazing.
yeah.
That's incredible.
Yeah, the scientist sitting in Science 2 seat did the ID, Virginia.
Wow.
Yes, that's right.
Yeah,
How did that feel in the moment to see something like that?
it was very cool. I mean, I don't know a rare species from one that's pretty common unless it's one that I've seen a lot. So just kind of watching the whole van just kind of stop and just go, hey, this is something really unusual. That's always really exciting
Yeah.
to watch.
Yeah.
Yeah.
Oh,
Very easy to get wrapped up in that too. It was a really cool moment.
I love that.
Well, the
Yeah.
first time that it had ever been seen was, I think it's like 2015 or 16
Oh,
with
wow.
the Okeanos Explorer during the capstone missions.
Oh my gosh.
So that was really cool, and we found out that it was a new species, and we got some really good footage of it. But at that time, they didn't have a suction sampler, so there's no way to sample that animal.
Okay.
Oh, wow.
Some very round rocks. You know, my other favorite geology term is botryoidal.
Ooh,
It's
that's
a good one.
It's a really good one.
Yeah.
I was writing up a little summary of what we saw from the previous dive last night, and botryoidal was used a lot. And it's going to be used a lot on today's report.
So what does that mean for those of us who don't know?
So that is a textural term, and when I use botryoidal in this context, I'm talking about the surface texture that we see on the ferromanganese crusts, and it's that very knobbly sort of bulbous texture that you see. And we see that develop a lot with minerals that precipitate out of hydrous conditions, kind of like this. So things that precipitate from water will sometimes adopt this kind of morphology. It looks like kind of a bunch of grapes all welded together. Yeah.
Oh, that's an awesome way to put it.
Isn't that where it comes from, like Greek?
I think so, yeah.
Oh. So the botryoidal, like, minerals or the shapes that come from, what causes that?
It just has to do with how the atomic structure of the mineral, or in this case, the crust, the rock, is arranging itself, and that can control what the surface, like what the crystal habit,
Yeah.
or the surface texture development looks like.
Oh, wow.
So, small scale translating to large scale. I mean, not all minerals do that though. So something like salt,
Yeah.
like typical table salt or sea salt, if you precipitate that out of water, I don't think I've ever seen that form a botryoidal texture. That's
No.
pretty much always going to be cubic
Yeah.
because of the way the internal atomic structure sorts itself out as it crystallizes.
That's incredible.
Yeah, so it depends a lot on what the chemical makeup is. And,
Wow.
you know, there are other conditions too where the crystallization pattern for certain minerals will vary as a function of condition, but
Yeah.
I haven't studied mineralogy in enough detail to understand some of the controls on some of those differences in morphologies. Yeah.
Wow.
I know it happens though.
Yeah. It's just so crazy to think, like, how old this environment that we're seeing is. You know, seeing
Yeah.
it for the first time.
So yeah, the ferromanganese crusts grow at a background rate of something like millimeters per million years.
Wow. Oh my gosh.
Yeah.
And those
There-
crusts are
there's-
pretty thick when you cut them open, right?
They can be.
Like, what's
So
the
they're-
thickest?
That would be out in the Western Pacific, where the seafloor zeoldes, seamounts are
Yeah.
on average quite old, and you can get them, inches thick.
Oh, wow.
The one that I was working on last night from the first dive, so we got that open. The ferromanganese crust ranged from,
Is that a something up there? Looks like a
what's it about?
something.
Ooh, I like something.
Oh,
like 2 to 3
There's a
centimeters.
shrimp.
There's multiple somethings coming up.
Yeah,
There's
there's
something on
something
the rock.
coming
All
up.
right.
multiple
Coral?
somethings.
There's a something. You zoom in video?
Yay.
Zooming.
Oh.
Is it a dead something?
Oh no, it's a live sponge. This is a
Hey.
demisponge.
Oh,
Ooh.
sponge.
But it looks like a
Pretty.
coral, doesn't it?
Yeah.
It does.
It really does.
You know what? We could do a Niskin bottle here. We haven't done any Niskin sampling yet.
Okay.
Okay.
I think this might be a good spot. Best spot
Let me
we've
get
seen so
planted
far.
here.
Okay, coming wide.
Yeah, you can see a bunch of feeding traces from like a limpet or something on the rock as well.
Oh, wow.
All right. Semi-planted. Can you pan and tilt digits on that?
Okay, zooming.
Oh, wow.
That is beautiful.
It's really nice.
Yes. With a shrimp there.
So that's a sponge, you said?
It's a sponge.
Wow.
Yeah, it's a demisponge.
Oh, wow.
Steve suggests Stellodorix sponge,
Yeah.
maybe?
Yep, Stellodorix. That is the sponge.
Okay.
What qualities about the sponge make it different from a coral? Like, how did you know it was a sponge, even though it was
Well,
coral-shaped?
the sponge doesn't have polyps like a coral.
Yeah.
But from far away, I could tell it was a sponge because of the color and the way the branches are sort of latticed together, where a coral wouldn't, make an intermesh like that.
Yeah.
Okay, I need camera action. So
Yeah.
Zoom out.
Zoom out.
Oh, wow.
Oh, there you are.
Oh.
You're all the way up there.
Yeah, unfortunately we're on a slope here, so I gotta. Hung up. Uh-oh. 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 Uh-oh.
Is the arm in a space it's not supposed to be in?
Yeah.
Yeah.
Okay.
That's not good. That's way not good.
Can you slew all the way over and use your sample camera?
Let's see, we need bubble cam.
Yeah.
Where is it? Do you have control over that here?
Yep.
Oh, do I have bubble?
Yeah.
I think, yeah. And right? Oh boy. Bubble's a little spicier. as I can go.
I'm fully hung up in the light bar. Holy cow.
You can't extend out?
I can't, I am hung up on something up there. I can't azimuth, I can't go down.
Oh.
Huh.
Try zooming in a bit on your Atalanta view to see if it helps at all.
Yeah, if we can, maybe if he can negotiate over that way a bit, so we can,
Okay, yeah.
let me come off the rock here. Well, here, I'll just drive over to it. We're just abandoning this thing.
Yeah, sorry, Annette.
Oh, no, okay. All right.
So
No
no
sample.
No Niskin.
Let's see what's going on there.
All right, I will hold position here, and then we're gonna.
Okay, you ready for me to zoom in on Atalanta?
Yeah, I'm gonna rotate around here.
Okay, and Levi, just so you know, I'm moving your lens in, so if you're not, just don't want to give you a startle. And then are you able to tilt up a little?
Yep.
Yeah. All right, yeah. All right, if you're driving, drive. I'm gonna move ahead a little bit. Can you tilt up?
Yep.
All right, more zoomy.
Okay, more zoomy. Looks, yeah, like you said, light bar.
Yeah, so
Yeah.
Yeah, but. Huh. Huh. What the heck?
Is something not responding?
yeah. Is one of your joints frozen?
Pausing 4 and 5.
I think it's a wedge in there. I can't.
Can't move it.
Oh.
Annette, are we passing multipuffer?
Yes.
Okay, got it.
I can't see what's holding it up here. Huh. Yeah. What to
All right,
do?
that's max zoom for me.
Hmm. It looks like he's just bumped up against it, but it's not.
Yeah,
Yeah.
Not
it
budging.
must really be stuck.
Robert, do you want me to find anyone else from ROV to help?
Let me just try restarting the arm.
Okay. Standing by.
While you're restarting, I'm going to go a little bit wide so you can get a positional perspective. All right, that's full wide.
Maybe not full, but
Okay. Well, just for Levi to know how
Yep.
far.
I'm gonna pull up a bit. I'm getting below.
yeah.
Okay,
What's
going
it
in a
doing
little
now?
bit more.
It's moving on its own.
It is.
Oh.
That was interesting.
Yeah.
Hmm. Dang it.
Okay. There you go.
Ah.
Hey.
Yay. Okay, coming wide on Atalanta.
That was weird.
Interesting.
They like got frozen or something.
That's
Oh.
weird. Yeah, it's full wide on Atalanta.
Thank you.
All right.
All right.
So are we planted? We're settled in, right?
Yeah, we're not
need
moving.
a bio break.
Okay, I'm watching. Oh, that's a shrimp. I was like, what's that behind us? Oh, see, the seat is so much warmer. What was
I like the idea of requesting Snuggies as official Nautilus gear.
Oh, yes. Have you seen the ones that have like a pocket and you can put your dog in it? And you can share, I was like,
I
I
love
want that so bad. Snuggies at modifications.
I mean, we can't have pets on board. We could put a rock in it.
Ooh. What if we put a rock?
That'll be your pet.
we should have pet rocks.
Put some googly eyes on it. That'd really make it.
There are
We do
googly eyes
like
somewhere.
googly eyes around here.
Yes, we do.
We need more.
All right. So, is there still interest in popping a Niskin once Robert's back?
Yeah,
sure.
I think so.
Yeah.
Okay.
Okay.
Alrighty.
Yeah, we'll
All
turn around and go back to those animals we were looking at.
Okay.
Yeah, it seemed like there was an interesting collection of different things there. So
Okay. Steve says we're good on any physical collections. He says the sponge has been collected.
Yeah.
by the Okeanos,
Oh, we collected some,
up in the monument.
I think last year as well,
Okay.
the Stellodorix.
Yeah. But yeah, Niskin would be useful here. Well, speaking of pets, my mother just sent me a picture of my cat snoring on her leg.
Oh, cats can snore.
Cats definitely snore.
Oh my gosh. That's amazing. Cute, cute. I wonder if do any sea creatures snore? Can any of them snore?
I think that requires air.
Is that like more like gurgling?
I think that also requires air, but more liquid.
Yeah. Oh.
Yeah, she's watching my cats while I'm away. So
Oh.
hopefully they're behaving.
Knowing cats, probably, maybe not.
And I've heard reports of one knocking things into the garbage can.
Oh, like trash into the garbage can, or just things into the garbage can?
Things that are elevated and can be knocked off.
So if it was trash, a plus.
She's just good at doing that when she wants something.
We have a question from a 3-year-old named Nova saying, what is a shark doctor called? Does anybody know what a shark scientist is called?
Ooh.
What was the name for
Would
that?
that fall
Fish
into
scientist?
Ichthyologist.
Was it ichthyologist?
I was
Just
thinking that would
under,
fall into
No, it's
ichthyology.
just under fish, right?
Yeah, I don't know.
Oh,
That's
yeah.
a really good question. We're going to
I'm-
have to go look that one up.
Yeah. Is it ichthyologist? I feel like that's such a big umbrella.
Yep.
Clearly we need to bring one on board for an expedition.
Right?
Yeah.
Oh, that would be really cool.
Well, I know one of our Atalanta pilots and also Duck
I'm going
Chief,
down.
Mike Burns, studied sharks, so he might know.
Oh, whoa. Oh, that is awesome. I know just the other day in Port Aransas off the coast of Texas, they caught a hammerhead shark that was 1 inch away from the world record
Wow.
of caught hammerheads. Yeah,
Wow.
it was
I'm
big.
getting a bit too close to the bottom.
They are called shark biologists.
Oh. I was like, where's the fancy name?
Yeah, so this is the spot where we're going to take the Niskin.
Is that a little Poliopogon?
Oh.
Ah,
And
That's
is that a
something there?
fish?
It is, I think it's a halosaur.
Okay. Ooh.
Yeah, they hang out like that. That's how I know what it was from far away.
Oh, right.
With their
sort of
head
tail
down,
up.
their tail up.
Yeah.
This is Aldravandia.
I'm full wide on the Atalanta view.
Oh.
And I'm not hearing you, Robert.
Yep,
Yes,
Niskin.
please.
We'll start from 1 and work our way down. So I'm gonna pan over here, see what you're doing. Okay. Gotcha. Okay, all the way back. The top one.
Did not see a floaty thing yet.
We can't see number one, right? Oh, yeah, the one closest. Oh, it's the one over there.
Oh no.
Oh, I guess we're putting it in the basket. All right.
Herc decided to wear a little jewelry.
Oh.
Can we just pop that into one of the forward bio
porch out?
boxes and
Yes.
deal with it later?
What is that
that's
on
a
it?
good idea.
What did it grab?
One of the Niskin rings
handles.
came off.
My porch isn't going out. Oh, it's all the way out. Okay, starboard box. Oh no, that's forward box open. Forward box. Sorry.
And this was sample number 38.
Oh. Is it?
Oh no.
All right, cool. Thank you.
Did the sample get collected though?
Well, we pulled the ring.
Okay.
Yeah.
Okay. And that was bottle 6. Is that correct? Or bottle 1?
Bottle
Bottle
one.
one.
Bottle 1. Okay. Thank you.
Oh no.
Is the ring just really on there? Yep.
I just got to get the right angle to wiggle it off. That's
Yeah.
all.
Oh.
Maybe.
Right, we might want to go back to the balls.
Yeah, balls can't get stuck on your hand.
No. I mean, I like the aesthetics,
There
but
we go. Perfect.
Okay.
the rings seem to be a little less practical.
Right. Forward box close.
Okay.
Oh.
It's not closing.
Is the bio box loose?
Oh.
Oh,
I don't
dear.
know. Okay, forward box open.
Oh.
Okay. Okay, fortune.
That's a new one.
Yeah, that's new for me. Well, maybe it's kind of. Maybe if you push it from the right side while I try to close it. No. Never had that happen before.
Right?
It's really, it must have jumped. Yeah, it must.
Yeah.
Cool.
All right, well, should we move on for now and
What
we'll
should we do?
try to mess with this for a little bit later?
Let's see what Bubble can tell us. Yeah, I can't.
Ouch.
Yeah, it's all the way out. I won't go any further. Should I put the porch out? Porch out?
It's not hung up on the plate. I don't know what it's hung up on. Oh, it's hung up on the camera.
Oh. Cool.
Oh no.
Is there any way we can wiggle it back into alignment with the porch all the way out? Okay. No, that's not what I'm suggesting. No, that's way too risky.
Yeah, I see.
I'm wondering if we could do it from the front corner somehow without knocking the camera.
Are you talking about the Zeus Plus camera or the
Cinema cam.
The cinema camera.
Got it.
Yeah,
I
we've got a
see.
great view of the bio box from the cinema cam.
Ah.
The black strip around the front of the camera, the plastic.
The hood, yeah.
Yeah, but it's hung up on the
Yeah.
shooting aluminum bit.
Yeah. And it's causing it to turn.
Mm.
Well, what should we do?
Do we want to keep moving uphill, box out, socks out for a little while?
Yeah.
I mean, I can't think of anything better to do unless we try to wiggle it a little more back into where it's supposed to be.
What if, could you put the arm inside the box and maybe direct it? Mm-hmm. Yeah, because I think it's this little metal piece on the side of the box is getting hung.
So do we need to try to scoot the box a little bit to the starboard side?
Oh, there we go.
Oh, hey.
Oh.
It got straighter.
Yeah, it looks a lot better.
It does.
Yeah.
Okay.
Yeah.
Yeah.
Oh,
coming
in.
in.
Yay.
Okay. Now
awesome.
Success.
We should look at that.
I don't think I like the rings on the Niskins anymore. That was the cause of all of this. Okay.
All right.
Back on track.
Literally. Oh, I'm so glad we were able to get that back in place. Thank you, Robert.
Oh.
All right, so what now? Now do we move? Steve's down there like, no.
No more sediment.
All right, since we're gonna get moving again, I'm gonna turn on 4 and 5. So they were paused.
Got it.
So these are the
Okay.
same ones that were running before.
Thank you.
So 10:59.
All right, so with about an hour left in the watch, we're just about halfway between waypoints 3 and 4. So we're making pretty good progress. It didn't feel like it for a while with that sediment field, but we're making progress.
Yeah. It was oddly peaceful though, the sediment field. Reminded me of some sand dunes.
For like 5 football fields.
Yeah.
Yeah, that's what I'm thinking. I put a note. Don't push the box out so far. Oh boy. That was my first forward box experience.
Same.
I'm gonna go up.
I'm looking over Giselle's shoulder.
Oh, did you
public
see
chat.
the
Somebody
joke?
says, what do you call someone who studies sharks? A fantastic researcher.
I love it.
Another person also commented, a pointy ichthyologist.
And yeah, you nailed the spelling.
Yeah.
Would a shark doctor be a surgeon fish?
Oh, I love that. That's so good. Oh, we have a good question down here. Somebody's asking, what is a Niskin bottle? We'll be throwing that word out a lot, right? Annette, do you want to take that?
Sure. These are bottles that they're open on both sides with lids that we can trigger on each side. So we use them to collect water samples. And so what you saw was Hercules' arms pulling the trigger to close the top and bottom to capture the water so that we can bring it back on board the ship. We're going to use that water to pump it through a filter to collect some supplemental eDNA samples. So this is the environmental DNA. We'll pump that over a filter, and then we save the filter in a freezer and bring it back to the lab to analyze. So we've got 6 Niskin bottles. We're triggering them at areas along this dive what seem like they have some biodiversity. so that they can be snapshots of the communities that we observe.
And what's the composition of eDNA? Like, what is that made of?
So that can be any kind of, for animals, it can be any kind of like pieces of cells that, or tissues that, cells that have sloughed off, tissue fragments that are generated from predation. That can be gametes, it can be fecal pellets, basically, you know, any kind of cells or materials that have genetic signatures of DNA in them. So we use that as like a genetic fingerprinting type of approach where we can sequence that DNA to determine who is living in the water without actually sampling the animals directly.
Oh, thank you, Annette. That was
Sure.
awesome.
We have our samplers running in the background, right, as we move along the transects with our survey. And then the Niskin bottles represent points along that transect. So the samplers are sort of integrating over distance, where the Niskin bottles are point sources.
Oh.
So,
Oh, wow. Oh, incredible.
So yeah, the difference between what we're doing with the multipuffer and with the Niskin bottles, we're getting eDNA from both, but the multipuffer can do that filtration right there on the vehicle, so we're doing it in situ. Whereas the Niskins, we have to do that filtering back on the ship, so we have to recover those water samples when we get the vehicles back on board.
right. Yep.
And is there kind of like a ticking time clock when it comes to collecting those samples from the Niskin bottles, or you can do that?
Oh, so when we when Hercules will come on board, we will process them right away.
Yeah.
We keep the water cold. We transfer, well, a couple of steps. We first transfer it into sterile bags, which we then store cold and process them right away because we don't want the DNA to degrade. So it's important that we work quickly, as quick as we can. The Niskin, the samples, the filters from the sampler will, again, we process those quickly as well, and they go, again, straight in the freezer is how we preserve them.
Oh, wow.
And so when the vehicle comes up, we still have a couple of hours of work ahead of us in the lab on the ship, right? So so our work is in some ways beginning when Hercules comes back on board.
Yeah.
So,
wow.
Yep.
Oh.
Yep.
Some good ocean forensics. It was awesome.
Yeah. Yep, the science team gets pretty busy right afterward. I'll probably cut open the rock samples tomorrow, whenever the schedule permits it, and do some processing off to the side. But I tend to stay out of the way of the more time-sensitive biological work that happens in the lab, because my samples can wait. So as long as there's another time, like the next day or later that day, depending on when the recovery happens, that's when I step in and I do my work.
Yeah.
Wow.
And then we also have people that are looking at the specimens that we collect, right? So we will often collect specimens of corals or other animals that will complement existing collections, and those are processed right away as well. So we're very busy on the ship, even when Hercules is not in the water.
Yeah. Wow.
I don't like that camera view.
This one?
The Niskin.
The Niskin camp? No, I don't like it either. It's really hard to see. It's very...
Yeah. Well, everybody was complaining about the other way from the forward looking back, and you couldn't see them all. You just can't. It's hard to see the whole rack.
Yeah.
You know?
6 is just too many.
Yeah. So maybe we need to come up with a wild harebrained idea to know when they're actually closed.
Yeah.
Beyond the camera view there. Need more cameras.
Yeah, we just need more cameras.
I support this.
You can never have enough
We
cameras.
need more screens, we need more cameras.
Yeah.
More
Maybe just
multi-views.
be like a fly and have compound eyes.
Yeah.
Yeah, get that 360 view.
That'd be arm hanging out in the breeze there. What's going on?
Just put a rail cam on so it can move.
No alarms stowed. What do you see?
Ugh.
It's all, yeah, it's all the way out.
Is it stowed? I don't think it's stowed.
You don't think it's stowed?
I think it's got its.
Is it flipped? We've noticed that it kind of droops.
Yeah. There you go.
Okay.
Are we just waiting for the swing to catch up?
Yeah.
Okay.
Oh. Maybe I gotta leave the hydraulic valve open.
Yeah.
Or it'll.
Yeah, it just gets all sleepy.
Okay. Carrying on.
Oh, there's a megamouth tunicate. See right here on the right?
Where?
Ooh,
Can we
Good
get
eye.
a closer look at that?
Yeah, right there.
You zoom in.
oh
Yeah,
wow.
thanks, Amy.
You have very good eyes. Oh my goodness.
Oh, wow. You do have good eyes. It's amazing.
It's amazing.
Oh. Can we collect it?
Oh, well, it'll
Right
just turn to
over
mush.
Well, yeah, yeah. Boy, that's too bad.
Looks kind of like a smiley face.
Yeah.
Really weird smiley face. Oh yeah, there you go.
Yeah, tunicates are weird animals. When they're larvae, they have a notochord, they can swim around freely, and then as adults they become sessile.
Really?
Mm-hmm.
Huh.
Oh wow. It kind of reminds me of like a jellyfish. Are they in the same family or genus or?
No, so the tunicates are actually in the chordata, so they're more closely
Oh.
related to us.
Really?
That's wild.
Yeah.
That is crazy.
So what's the sort of orange-red structure that we see in the center there?
I think that might be the gut.
Oh.
Mm-hmm.
What kinds of things do they usually feed on? Are they like filter feeders or?
Yeah, so they filter feed through that big mouth opening. They have siphons that they can pump water into their bodies with,
Whoa.
and so they'll just capture whatever comes by.
Megan, do you think they're more unusual to find in these surveys than other types of like sponges or corals? It seems to me that we encounter them less often.
It depends on where you are. They're
Okay.
not particularly rare. I do
Okay.
see them quite often, but they are hard to spot.
Oh.
Okay.
So,
Okay.
yeah, you might not see it right away, and you might
Right.
pass over it because of how
Sea
transparent
cucumbers?
they are.
Yeah. The camouflage is great.
Oh, there's a nice purple sea cucumber.
Ooh.
It's a nice juicy one.
It's like a sweet
Kind of like
potato.
a shadow, doesn't it?
Oh wow, look at that.
Can you zoom
Yeah,
in?
they can get some pretty
Happy
vivid
zooming.
colors sometimes.
They are.
It says sea cucumber is in the family Synallactidae.
Look at that.
Yeah, I think we saw a couple of these on the first dive.
Yeah.
Yes.
Yeah.
Yeah, this might be a Benthidites.
These guys really make me want a purple sweet potato.
Yeah.
Or an
I
eggplant.
do love
Ooh, yeah, an eggplant too.
This is like an
I'm like,
eggplant.
ooh. We're back on the sea salads.
Just watch, it'll be
Sea
eggplant, eggplant parmesan on lunch, right?
Oh my
this one likes
gosh.
them for us.
Oh.
It's doing the
Ooh,
I sense something movement.
Yeah,
a disturbance in the force.
Maybe we should back away
That's
from
called
it
the
then.
water.
All right, carry
All
on.
right.
How cool.
More dead sponge pieces.
Yeah. Is that what those are? Dead sponges?
Yeah.
Oh.
Yeah.
It's much easier to center the camera with the lasers on.
Is it?
Yeah. See, then I can just make the lasers go in the middle of the view.
Ah.
And I know how big the animals are. Win-win.
Yeah.
It is a win-win.
Oh,
Well, the
wow.
lasers are on the camera, though. I don't know what you're saying. Center with that view? It's always in the center of the view.
No, it's not.
What?
Watch. Oh, I guess it is.
It makes it really easy to center the camera.
Yeah.
The camera. He's always in the center.
Yeah.
Oh boy.
If they're off center, then we've got a real problem, I guess.
It could be true if you could see it in that view,
Yeah,
but it's hard to see.
it is hard to see it in that view.
Yeah.
The green is hard.
Can you zoom in on that?
Zoom in on the
The
top
upper?
camera,
Yeah.
the down camera.
With all your handy dandy
Upper,
controls.
upper,
there?
yeah, like zoom in. Oh yeah, I totally can. Look at that.
Ooh.
So many buttons. I found a new button though.
Oh no, what is it?
It says self-destruct.
Ooh.
That sounds dangerous.
Yeah, it's on the bubble chem page. I was like, oh, we don't need that button. We're
Do you have
already
to put
having
in like
enough.
a Star Trek
adventure.
style? Does it require Star Trek style authentication code?
Probably.
So we do have a button.
I like the button. The button. Where's the buttons?
You gotta have the button engaged. And I've been told that I'm the one that requested the button.
It sounds like something that you would request.
No, I edited the code to take the button out, and the guy who wrote the code came out the next cruise and put the button back in.
It sounds like a button battle.
Button's silly. Don't need it, that's only button.
I was telling other Levi about the button. Gave me this weird look about it.
So he doesn't think it's necessary either?
No, it's definitely not necessary. It's dumb. We have another, but this is a dumb button
Oh.
too. This one. This enables these. Without that,
Oh, button. Okay,
these
can't-
don't do anything. Like,
Yeah.
why?
So people can't accidentally push the buttons to change your
But
Z
it's like,
bias.
you always have, there's never a time that's not engaged.
So for anybody listening in live, back row has no idea what they're talking about either.
I don't know what a button is.
Yeah, we just got to
Buttons.
like
We're just along for the ride here.
So we're talking about pilot interface things that have been the way that they are for years and years and years.
Because
Yeah.
that's the way it's been.
That's the way it's been.
And they stay that way because they've always been that way.
Yeah, they've always been that
yeah.
way.
I understand.
Every
the
vehicle
legacy
has
buttons.
its quirks, you
Yeah.
know.
Mass
Yep.
spectrometers have some of those too.
Yeah.
Do they have buttons?
New plasmas have a big red button that you never push unless you absolutely have to push it, but I've
Don't
never
push
had
it.
to push it.
Well, we have one of
Ooh.
those too, or
Yeah.
a number of those. We have several of those.
Big red
actually.
button.
Good, I'm glad.
It's not a real system without at least one.
Yeah.
Yes.
You gotta have
Good
a
big
red
red
button.
button.
Yes.
So big moving mechanical bits need that. And I imagine
Yep.
a mass spectrometer has high voltages and bad things happening.
Yeah.
Yeah, it's pretty typical that those are running anywhere from 6 to 10 kV for the kind of
Yeah.
work that I do, and then there's some
Yeah.
others that go even higher.
Yeah,
Yeah, big red buttons are very important.
yeah.
On this rock here, there is
Reaching.
a thing. Right, that small
The
lonely rock. There's a thing on it.
The lasers are almost on it, rock, right there.
Yeah.
Okay, can we zoom in?
Oh.
I
Copy,
hope Steve is
zooming.
watching. I think this is a black coral.
Ooh.
Ooh,
It looks
a
like
very small one.
a heteropathes.
Wow.
And I think there might be some interest to collect it.
Ooh.
You have such
Okay.
a good eye.
are we able to set up for a snip then?
Oh, look at that.
Hang on, I'm gonna get Steve in the
Yeah, see what
chat.
Steve says. Oh, and there's
Are
a
we-
little tiny
Are we
amphipod
stopped?
on it.
Are we still?
We are stopped. We are not moving.
Yes, Fanny confirms collection would be good. She says we could take the whole thing.
It's the whole thing.
Take
Yeah.
the whole thing.
Is this a snip and slurp?
I think it'll go through the slurp.
I think so. It's small enough, right?
Yeah.
Yeah. Maybe bottom first so it
Oh.
has less of a chance of getting stuck in the hose, if possible.
Annette,
Oh.
are we pausing the pumps?
Yeah, I think
let's
we
see,
can
we
do
need
that.
porch out.
Okay, pausing
Porch out.
4 and 5.
Good call, because we just kicked
Copy.
up some sediment. We're above it right
Yes.
now, though,
Yeah.
so you should be good.
They're off.
And right. Tilt down.
All right, so this will be sample 039.
039.
All right, lots of zoomage.
All right, zoom in.
It'd be really cool if we get that amphipod too. But I think it might swim away.
Hang on. A little less zoomage.
Less zoomage.
Which mode are we in here? We're in that mode. All right. All right, more zoomage. Oh, you got away.
Yeah, I
Yeah.
was pretty sure it was going to swim away. All right. Should we switch to the sample salvo?
Oh,
Oh, nice catch.
Good
Hey.
Levi's, can you switch to the sample salvo?
Yes, sorry. Yeah?
Where's Those are
Slurp.
Slurpees.
You gotta turn the camera on. So, Levi, you gotta do this. Go hotel cameras, rail off, bucket on. Number 1?
Number 1 sounds good. Okay, section
Alright,
is
I
on.
need some more zoomage.
Okay, zooming.
Oh, let's turn off. Oh boy.
Oh,
We're
dear.
sucking up dirt.
Yeah, we're in the
Oh,
dirt.
no.
I guess we have
I
a
gotta
bonus
get up
sediment
off the
sample.
bottom. Did you
Good.
kill it?
I killed it. It's off on mine. I don't know
bad.
why it's still going.
Digging a hole.
Okay, bonus sediment sample. We'll handle that in the lab.
Are we gonna go to a new jar? Oh, dang it. Get up.
Yeah, let's go to a new jar. Or we can flush jar 4. I could, now that we're in the water column.
All right, go for it.
All right.
Okie dokie.
Okay. Now
Yeah,
you can put it
they
in
just
there.
flushed it.
That is number 1?
Number 4.
4.
Number 4.
Number 4. Got
It looked like a 1, but that was just the lights.
Yeah.
That's, I was confused.
Oh,
Number
no
4.
worries. It does look like a 1.
Can we, is that all the way over?
Yeah, there we go. Here's down.
Okay.
Okay,
Yep,
there you
yep.
go. I think you can let it go now.
All right,
Yes.
it's in.
It's in
Excellent. Good sampling. Thank you, pilots.
About that.
there.
All right, fortune.
Fortune.
And then do you have to rotate jars or I don't know.
Yeah, we can
I
get
don't see
rotate.
it yet.
You got jar rotate? You don't have jar rotate. Yeah, you do.
I have jar rotate.
I think you have all the functions. It's gonna be starboard.
I have box open, close, porch in and out.
All right. All right. You don't get everything.
I can't jar rotate.
No button, right?
Only self-destruct.
Okay, we'll go to flush, right?
Yes.
Oh, gosh. Annette, are we resuming multipuffer?
So I think we're actually going to start with another pair of pumps. So,
Okay.
Okay.
If we're ready, I will turn on pump 6 and 7. You got
Copy.
that?
Yeah, 6 and 7.
And 7.
Yeah, we're at waypoint 4, and it's going to get locally a little bit steeper for a while as we get into waypoint 5, and that slope
Okay.
break makes some sense to switch samples, I think.
Great. Yeah, I just turned it on. Thank you.
Right, and those are samples 40 and 41, correct?
I'm getting pretty close to the ground,
Awesome.
but the delta's pretty...
It's pretty low. You can come up. Right?
Yep.
All right, coming up. And I'll come towards you. Okay, everybody's
Okay.
happy again.
Great. I like when you're happy.
My brain for a split second there totally mistook one of the laser beams in the water column as a bamboo whip.
Oh.
Yeah. Yeah, that happens occasionally, and I always have to take a second and go, okay, beam or whip coral.
Yeah,
Right,
sometimes
so,
I mistake rocks for animals, so, you know, it
Sometimes I
happens.
mistake rock shadows for animals. Right there with you.
162? Is that our bearing?
Well, 245.
Or 24? Rat.
162.
What?
God,
No.
let's just go wrong the other way.
That was yesterday.
Yeah, that was yesterday.
Would you like the volume sampled for 4 and 5? Okay, so sample 4 is 177378. Sample 5 is 167209.
167209. That's a good number. There's a sponge on that rock. Straight ahead, right at the top of the screen.
Oh.
Oh yeah, sure enough.
Oh.
So just a heads up, front row, the slope, it was a little steeper. I wasn't sure what to expect here, but I'm seeing some little piles of cobbles, so I'm just kind of keeping eyes open for something that looks like a potentially good geological sample. I haven't seen anything yet, though.
Okay.
Right. Zoom
Yeah.
in.
Copy, zooming.
Ooh.
This is a glass sponge called Poliopogon. It is in the family Pheronomatidae.
This one is a pretty small one because they can get quite large,
Oh
can't
yeah,
they?
they can get huge.
Oh yeah, like ROV sized.
Really?
Yeah.
Wow.
What?
We've seen that up in Papahānaumokuākea.
Oh my gosh.
Yeah.
That's incredible. How old would that sponge be if it was as big as Hercules?
Oh, probably pretty old, but sponges are hard to date. They don't have any parts that are easily dateable.
Oh.
Yeah.
No social skills.
It's literally,
Yeah.
I can relate to that sometimes. Yeah, it's because they're made out of largely out of silicon. Whereas with corals, it is possible to get a better idea of how old they are because their skeletons are made out of carbonates, and you can trap very tiny quantities of radioactive elements like uranium and thorium in them, and we can take a look at something called the uranium series disequilibrium. So there's a whole decay chain that uranium decays into as it decays to lead. And if that's in disequilibrium, we can measure different parts of that decay chain and back an age out of that.
Oh, wow.
And that works for an age range of a couple 100,000 years, give or take, maybe a little bit longer. I can't remember off the top of my head. But eventually you do get to a point where the material gets old enough that no longer works because everything's in a state called secular equilibrium. So that radioactive decay is still happening, but all of the ratios and everything along that decay chain, you know, all the different proportions of those different products in the decay chain have reached a point where they don't really change anymore. Just they've all kind of evened out.
Oh, wow.
Yeah.
Oh.
It's a tricky thing to do. There's a lot of chemistry and very sensitive measurements that you have to do in order to get that age and measure it precisely.
Wow.
Yeah.
That's incredible work. Oh my goodness.
It is. So it's not one of the things that I do much of, but I've dabbled in some parts of it before.
Wow.
Yeah.
I know as a kid I usually heard about like radiocarbon dating. Didn't really fully grasp how it worked, but I always thought it was just something so cool that that's what scientists are able to do.
Yep, that's based on some cosmogenic nuclides formed, and exposure to atmosphere will mean that you incorporate some of that that cosmogenic carbon isotope, and you can measure that relative to stable carbon isotope. And if there's still enough carbon-14 left in the sample, you can figure out, based on the ratios of those, about how old your sample is. But carbon dating can be a little bit tricky.
Yeah.
Because there are multiple ways to make carbon-14.
Yeah.
And some of those can be anthropogenic.
Yeah.
So, yeah.
Wow.
What time were we covering?
8 p.m., right?
This evening, let's see. At 8.
Okay.
Oop.
Self-destruct. I don't know what that. You been looking at those? You ain't been looking at those.
Okay, so these ones will change that one. Cool. Good to know.
Not the self-destruct.
Not the self-destruct.
Oh, it does self-destruct.
How does it self-destruct?
Oh, were you changing your views on the BTZ there?
Hopefully we never find out.
Those are the
Right.
bubble options.
I'm like, is it like Lego pieces coming out?
Self-destruct.
Yeah.
Because you're looking at the J-box?
Oh, it's because you could get the arm hung up back there and like rip stuff out.
Oh, okay.
That makes sense actually, kind of. I think that's the thinking. Yeah. You might need to start looking at gauges and whatnot. You need some more training.
Yeah, and filling out the spreadsheet.
Yeah, you're dropping the ball over there. But important job is keeping the vehicle off the bottom.
Yes.
Mm-hmm.
They're all important jobs, but that's
Yes.
primary important job.
There's a lot of important jobs.
Yeah.
Mm-hmm.
Sitting in the front row is a multitasking time.
Not as bad as the submarine, though.
Well,
know?
that's because
Oh,
it's
I
just
can
you.
imagine.
And you
Oh
can
my gosh.
And you got to keep people alive.
Yep.
That's stressful.
Oh my gosh.
I like all these little baseball rocks. You just
Yeah.
want to throw them.
What kind of story does that tell about? They have a lot of like baseball-shaped rocks.
They do a lot of rolling.
Oh.
Hard to say. These could potentially be nodules. They could be highly rounded cobbles that rolled down from much further away. I don't know.
Yeah.
Yeah.
Another mystery. Ooh.
Yeah. I'm keeping an eye open for something that looks angular. I don't think we'll see a whole lot around here, but I'm
Wishful.
looking.
Yeah.
I'm checking it out.
Wishful thinking.
We had a really good question from the UK. They asked, given the number of unexplored seamounts in the area, how do you decide which features will be visited on an expedition? Is that like a multifaceted kind of question there, or?
I can take that one because I've been involved in the dive planning. We do like to try to favor seamounts that haven't been explored in the region, in the target region that we're looking at. It's really tough to decide exact sites, though, especially in areas like this where there's relatively very little data.
Yeah.
Here, yeah, this seamount was nice because there is already some map data that we can take advantage of, because that reduces some of the time that we have to spend on site doing map surveys.
Yeah.
And we can just plan the dive, get here, and launch the vehicles like we did this morning.
Yeah.
So that was one of the things that made this an appealing target. It's also a little bit of a different feature than what we dove on for the first dive, because it's a much smaller seamount. It doesn't look like it was ever above sea level. So it's different morphology, a different environment, different depth range that we can look at. So it gives us some different science goals that we can look out for.
Yeah.
Yeah, it's a really tricky kind of multifaceted question, and the answer can depend based on the scientists aboard, the kind of science goals that we have.
Yeah.
It's not, I know that's not a very exacting answer either.
Yeah.
Yeah.
But overall, lots of different things kind of come into play when it comes to selecting that site.
Yeah.
That's so cool. So truly kind of highlighting the fact that you need a team to make this,
you really
things like this happen.
Yeah, because it's a constant conversation that we're having in the science team in order to make these decisions. You know, we're talking about it with the operators, with the expedition chief. Yeah.
Wow. What?
Incredible. So here, this is a lot of really basic reconnaissance work that we're doing.
Yeah.
This is the first time we're seeing this seamount, and just getting an idea of what's here, what the geology looks like, what kind of communities we're hosting.
Yeah.
And just trying to understand the area better, and it's the dynamics of the story that it's telling.
Yeah.
Yeah.
Wow. I really like this up top view. Kind of reminds me of a chocolate chip cookie a little bit.
Yeah.
You're hungry.
Yeah.
It is lunchtime.
It is almost lunchtime.
Hershey's bar with the white chocolate and the cookies.
Oh my gosh.
Yeah, we are in the last 20 minutes of our watch, so 12 to 4 will be coming up in probably the next 15 minutes to come kick us off so we can go eat.
Yep.
But yeah.
Or take a nap.
Eat first. Eating is important. It's one of the things I've learned. If I go to sleep and I'm also really hungry, it's usually not a great morning.
Yeah.
Yeah.
I hear it's also good for like seasickness, right? To kind of help it stay away, right?
It can help to keep your stomach a little bit fuller.
Good tactics. Good tactics.
Yeah. Also, if you go too long without eating, that could be really hard on your stomach too.
Yeah,
sometimes that
I've
makes you
heard
feel
that.
sick. So keep that food in that belly.
Yeah.
All right. So once we're in a position where we can sit down, I'm thinking about picking up one of these cobbles.
Really?
Yeah.
Okay. Are we in a position where we can sit down?
Yeah, let me just hold position. Bridge now.
Yeah, because we've got some stuff that's a little more triangular.
Hey, can we hold position here? Thank you.
All right.
Yeah, I mean, these could be nodules, but we're seeing a lot of them, and they're all different sizes, so I'm kind of thinking...
Hard set on any particular one, or just the one that decides to come with us?
Big one, big one.
I'm looking. Let's see, so That one right there in the center looks pretty good.
Oh, yeah. Okay.
I need to see the front of the basket. See what I'm doing. See what I'm doing? That's good. So you were saying something in here?
Yeah, the one right
so
up.
that looks interesting.
Okay.
Yeah. It's a nice size. A little over 10 centimeters. You know, it's something different here, but still rocky, so
I'm gonna pause the multipuffer.
All right. Yeah.
Was this one here?
I don't think, yeah, that one.
Look, it's loose. Got the creeps. I need to calibrate it. I don't like it.
It's a little fast.
Oh, it's got the creeps too, though.
Yeah.
This boat, it wants to keep going.
If that one's not easy to grab, maybe we can get the slightly bigger one in the bottom left. Okay.
There it is.
We're just goofing with the arm a little bit.
Okay, yeah, I like that one.
Very round, though.
Yeah, it does.
What do you like about it?
I'm curious what it is, because this...
That looks like a...
It could be a chunk.
Cannonball.
Yeah, I mean, chances are there's some sort of lava in it, just really weathered.
It's a sweet potato.
Yeah. It's not like the flat potatoes that can be a problem, but I like it. So
All right.
Ooh.
I think we'll take this one. Yeah.
Yay. It's rock o'clock.
It is. I'm not sure what we're gonna get with this one, but we're seeing a lot of these around here. So I'm breaking my own rules a little bit.
Uh-huh. Is curiosity winning over?
I think a lot of these roll downhill.
Yeah.
I think that's what we're seeing. It's a steep area.
Yeah.
So these aren't screaming like nodules to me.
Yeah.
So if you look here, this is also steeper because the contours are closer together.
Yeah.
Where you want this?
So I think this
A
is
sounds
kind of
great.
coming down here, and they're more rounded because they've tumbled more and they've broken off a bit more. That's the sense I'm getting.
Yeah.
It's truly so amazing how much of a story
Thank you
rocks
very
can
much.
tell. Yay!
And that is 042.
042.
Okay, back to you, dive, whatever. Moving on.
All right.
Bridge now.
Are you restarting the multipuffer? Okay.
Hi. Let's do
Thank
30
you.
meters, 245. Perfect.
Alrighty.
All right. Thank you. Yeah, so this is one of those situational things where, yeah, the environment is doing something different than it was earlier, and there are situations kind of like this where it's like, okay, what can we learn from what's happening here? So that's why it's worth grabbing a sample. And if there's not a lava inside, then yeah, we've got something else going on than what I think right now, which I think this looks like rocks tumbling downhill from somewhere further up.
Mm-hmm.
So if
There's
it's
some little
something
white-
else like a nodule or if it's a sedimentary rock, then yeah, I'll revisit that
is there
because
white
that means I need to my interpretation right now may
I
not be
don't
correct.
see anything.
Yeah.
Yeah.
Wow.
It's just sediment stuff.
Yeah.
So I'm not always a hard rock geologist. Sometimes it's sometimes you
White
need to know
sediment.
more about the context.
Mm-hmm. Get that
I
full
think this is a
picture.
good place to
I
do
gotta
that.
look up
Yeah.
there more often.
Yeah. I really am a big fan of this landscape, though. I think it looks so neat.
Yeah, yeah.
Mix of sediment with some rounded stones. It's so calming.
So yeah, these look like cobbles that have traveled a fair ways downhill.
Yeah.
So might be right, might be wrong.
Someone said all these rocks here would
Okay.
make great pet rocks, by the way.
Yes.
To crochet them little hats.
Crochet
yes, we
them
should
little
crochet them
hats.
little hats.
Megan could do that.
Yes. Every rock will get a hat.
All right, so over the next few minutes, you'll hear some new voices start to trickle in. You'll start to hear us change over watch and go on and off mute. So if you excuse our brief chaos, we'll get things changed over the course of the next 10 minutes.
Yep. Thank y'all for tuning in. Stay tuned for what we find next.
Yeah, and we will enjoy this very rocky, cobbly space while we do that.
It's so cute.
I wonder what's for lunch today.
Right?
I'm ready to eat.
Mm-hmm. The chocolate chip landscape. There's just something about naturally tumbled stones. I love them.
Oh yeah?
Yeah. Like I just think of how much time must it have taken for these to have rounded.
Depends on the
To how-
composition of the rock and how
Right.
hard the rock is.
Yeah. And like, was it before it was covered in manganese crust or after?
Before.
Before?
Yeah. Otherwise, you'd see more evidence of like corners through the crust. They'd be rounded over because of the crust, but you'd see more angularity to it.
Oh.
Here we don't, and that's kind of cool.
It is. Kinda it is. Yeah.
Yeah. Yeah, I'm here primarily to get the lava samples, but my sense is that these have a higher likelihood of being lava samples than I normally would think for a very rounded rock.
Yeah.
So it's a trade-off. Also, with this one, there was never any carbonate cap or anything.
Yeah.
So I'm not worried about picking one of those up.
Yeah.
So this thing this seamount was never large enough to make it above sea level and like start developing reefs.
Yeah.
Yeah. So it should be some mixture of encrusted lavas, plus or minus things like manganese nodules and maybe some sedimentary material.
Yeah.
Yeah.
Wow.
Okay.
Incredible.
Well, I'm going to start working my way out of the watch lead seat, so Steve can take over, and then we'll get things moving again here shortly. So I'm going to sign off. I think we'll be recovered by 8 o'clock, so you might not hear 8 to 12 again today. We might cover the very end of watch depending on what happens. So,
Yep.
we will see you next episode.
It's 12, signing off.
Video watch change.
Front row, are you still getting situated up there? Okay. No, that's good. Yeah, let's take a look at it.
Hi everyone, this is Science Communication Fellow Matt Holden. We are going through a shift change, so we will be right back with you in just a few minutes.
Yes, sir. Oh yeah, absolutely.
Yeah.
Okay.
We've seen this a couple times over the course of the dive so far. It's umbulella, but this is the best image we've got so far.
You want full zoom or stop there?
We can go full zoom since we're still waiting for.
And Steve, you said that's called umbulella?
Yeah, it's a sea pen.
Sea pen.
Yeah, it's a type of octocoral. This one has 4 polyps. Usually they can have several polyps on the top.
Very cool.
But it's a typical deep sea pen. It's not found in shallow waters.
Okay, awesome, very cool. And they use those polyps to catch their prey?
Yeah, the polyps have tentacles that are filled with nematocysts. So these are stinging cells that will capture either other animals or particulates out of the water column and ingest them. And actually you can see the gastrovascular cavity, which is the darker red maroon color in the center of the polyp. So that's its gut and stomach.
That's awesome. So cool.
All right. Yeah, we can take a look at that.
While we're moving, I just wanted to give a shout out to the Baird family in Massachusetts. They're watching us right now. So, hi.
Hello, hello. For those of you that are just joining us, we are on dive number 2 of expedition NA174 to explore the deep sea waters surrounding the Republic of the Marshall Islands in search of biological and geological samples. We are currently at a depth of 2,562 meters, and some of our major goals on this specific dive are to, again, search for new biological samples, possibly new species or new range extensions. We will also be collecting some eDNA or environmental DNA, which will give us a better overall picture of what organisms live in these environments.
You good with that, Steve?
That's great. It's awesome. So this is a flytrap anemone,
Whoa.
also known as Actinoscyphia. And it's living on top of a long sponge stalk. The sponge is long dead, but we typically find these anemones and other filter and suspension feeding organisms like some barnacles a bit further down the stalk, associated with higher parts of the terrain, and any way that animals can get up and off the deep seabed into the flow of the current is a good spot for them to feed. All right, really nice photos. I think we're ready to move on with our move.
You said this is a type of anemone, Steve?
Yep, it's a type of anemone. Actinoscyphia.
Amazing.
It's a particularly large one, but you find this genus globally.
And they're always in the deep sea?
Yep, yep.
So you said that was a
Is
large...
2.45 fine?
Can they get... How big can they get?
That's about the average size of them, but they can get a bit bigger.
Okay.
Yeah, depending on the situation, where they're sitting on. Sometimes they're on the seabed, sometimes they're higher up.
Ridge now.
They will often parasitize any substrate. So if there's a coral that has a bare part of its branch,
Okay, can we step
they'll
30
settle on that, and they
meters
can
at
live
8.45?
parasitically with the coral
Gotcha.
branch. You find them all over.
Will they eventually end up killing the corals if they live on those branches?
They can grow large enough, but they typically won't kill an entire coral. They'll just grow larger. But corals will also have mechanisms to defend against such types of parasitism.
Okay. Could you explain some of those mechanisms?
Yeah, they're really interesting, and they're kind of rare to see, but we're starting to see them more and more, especially in the deep sea. But in shallow water, corals have a lot of different defense mechanisms, like the ability to defend chemically against other organisms in their surroundings. So they might, put out a distasteful or noxious chemical to other animals that might take their spaces. In the deep ocean, though, things tend to grow pretty slowly, and chemical defenses are still present in things like sponges. However, these types of deep water corals, for example, might create cysts, like a tree might create a gall around, a wound or a parasite.
Yeah, definitely.